Hello friends, this is Sonu. As you all know that we both are discussing the pharmacology, me and my friend Subham. So we have decided that some other medical subject also should be discussed. So from today I'm going to discuss about very important medical subject, that is the microbiology. But along with the pharmacology and microbiology, some other useful subject also we are planning to discuss. It can be such as like some important subject, biochemistry, pathology and physiology, histology and so on. So very many important topic we will discuss about some biochemistry and some pathology and so on. So from today onward I'm trying to discuss the microbiology. As you know that microbiology is a very important subject in a medical student. That's why I'm going to discuss about the microbiology. So first of all, what is the microbiology? Microbiology, you can see that micro means very small. Small. Macro means big, large, and micro means small. Bio means living organism. Living organism. And logy means science. So we can see that microbiology means the study of the microorganism, or we can say study of the living organism that we can't see by my own naked eye. We have to see by the like microscope. Microscope can be, it can be electron microscope, it can be fluorescent microscope and so on. So we can't see by my own naked eye. That is the microbiology. So again we will repeat the definition. That is the microbiology. It is the study of the microorganism, or we can say study of the small living organism that we can't see by my own naked eye. That's why we have to see by the some magnification, that it can be microscope, light microscope, fluorescent microscope, dark microscope, and an electron microscope and some other microscope. And this is our main classification that we have to know about this classification. Actually microorganism is divided into non-infectious microorganism and infectious microorganism. For example, this is the marker. In the marker, lot of microorganism there. In our skin, lot of microorganism is there. In our nail, our palm, a lot of million microorganism there. But they are not infectious. They are not harm for our human body. Some of the microorganism can be tried to harm, but our immune system, our skin protective layer, they protect from them those microorganisms who want to try to inside our human body. So we can say those microorganisms are harmless. They are not harmful for our human body. And so we will discuss those microorganisms which is harm for our body. Those microorganisms which try to initiate the problem or try to start the pathology for our human body. So those microorganisms we will discuss. First of all, you can see this is the our very basic classification of the infectious microorganism. So let's start from the bacteria. First, this is the bacteria: fungus, protozoa, helminthes, ectoparasite. Like it's an insect like it can be lice, horsefly, and some other. From the insect, you can understand that which types of microorganism are belong from to the insect, and virus and prions. So from the bacteria, as you all know, that is divided into two parts. First is categorized from prokaryotic, and second is the eukaryotic. This also we studied in 10th, 12th and 11th. So eukaryotics belong to fungus, protozoa and helminthes. These three and ectoparasite. The four microorganisms are belong to the eukaryotic categories, and all bacteria, all types of bacteria are belong to the prokaryotic. So difference between eukaryotic and prokaryotic, you can see it's a very briefly discussion, very briefly difference between prokaryotic and eukaryotic. First of all, prokaryotic has singular circular chromosome. Means, for example, this is bacteria, and inside the bacteria there are gene. So this is a circular chromosome. These types of chromosome in the prokaryotic, means in the bacteria. But in an eukaryotic cell like fungus, protozoa, helminthes, ectoparasite, and as well as our human cells and plant cell also made by the eukaryotic. This is belong to the eukaryotic cell. So in our human cell, let's make our human cell. For example, this is our any human cell. Inside the human cell, the chromosome is the linear form, like that. These types of chromosome inside the eukaryotic cell, eukaryotic microorganism. Next difference: no any nuclear envelope and nucleolus in the prokaryotic, but in a eukaryotic have membrane bond nuclei present and nucleolus also inside the nuclei. No membrane bond organelles, but in a eukaryotic present organelles. Organelles mean it can be mitochondria, endoplasmic reticulum, Golgi apparatus, endosomes, and lysosomes, but these organelles are not present in a prokaryotic. Next, no plasmamembrane. Eukaryotic have plasmamembrane. It's replicated by the binary fusion. Only bacteria can be replicated by the binary fusion, but in a eukaryotic cell, it replicated by the meiosis or mitosis. You can remember. The prokaryotic have lack of sterol. Sterol means we can say is the group of the cholesterol. Means cholesterol are present in the our human cell, our human body. So actually the sterol, it's a, we can say the sterol is main function of the sterol. It's a prevent the osmotic burst, like the it will change the whenever change the osmotic pressure inside extracellular or intracellular can be burst. So this sterol are prevent for the burst. That's why prokaryotic cell are automatically burst due to lack of the sterol. But in a eukaryotic cell, our human body, in a human cell has cholesterol. This is the group of the sterol. And except fungus, only in a fungus has ergosterol. This is the exception, and protozoa and helminthes and ectoparasite don't have any sterol. So just in a fungus has sterol, and the human body cholesterol. The next one, cell wall are present. In prokaryotic, that is the bacteria. Bacteria has cell wall, but in a eukaryotic, they don't have any cell wall. Means absent cell wall, except fungus. In a fungus have, but this cell wall is not made by the peptidoglycan. Actually, in this case, in this condition, this cell wall made by the peptidoglycan. We will discuss about details about the bacteria cell wall. Just right now, I'm very briefly discussing because this is the introduction of the microbiology. So in a fungus, this cell wall is made by the chitin, and this is the pleomorphic. It can be changed the shape. It can be changed the on shape. It's not a specific shape. So the fungus is made by the chitin body. So that's why, except fungus and almost all eukaryotic cells don't have any cell wall. The size approximately 0.2 to 2 mm by diameter. This is the bacteria size, and about the eukaryotic size is depend upon the approximately 10 to 100 mm by diameter, but it can be increased depend if you talk about the ectoparasite. So it's depend about the which types of insect. Maybe it can be 200 mm, it can be 250 mm. It's depend upon the parasite. So this is our main, we can say, basic difference between prokaryotic and eukaryotic. So let's discuss first about the bacteria. So according to according to structure, bacteria is divided into two parts. First, typical, and the next atypical. Typical, first of all, we'll discuss atypical. In atypical, it is the mycoplasma, then rickettsia, and chlamydia. Actually, this is the, we can say, the group of the bacteria in the mycoplasma, like the, for example, mycoplasma pneumoniae and so on, rickettsia and chlamydia. These three are belong to the atypical. Atypical means the lack of the structure wall. This structure not like the typical bacteria, and that except these three, all bacteria are belong to the typical. All bacteria belong except these three. So mycoplasma, rickettsia, and chlamydia. This is the genes we can. This is the genus, and this genus, a lot of bacteria are which belong to this three genus. This is the lack of the structure wall. The original structure wall, that is the which made by the peptidoglycan. About the structure wall we will discuss. So this structure wall is not fully we can say fully developed like the typical bacteria. That's why this is three exception. This is belong to the atypical. The lack of, due to the lack of the structure wall. So according to the structure classification, this depends on the typical, atypical, and according to the staining, because a staining method means how you will recognize that this bacteria is belong to this group and this bacteria is belong to this group. So usually according to the staining method, because staining method is very important, then you can recognize that this bacteria has this types of properties, and whenever you will understand that this bacteria has this types of property, then you can very easy to treatment to patient. If the any person suffering from, for example, streptococcus, so if you don't know about the morphological structure of the streptococcus, the staining method of the streptococcus, then you can't think that which types of drug we should apply. That's why we should know everything about each bacteria. So for example, the bacteria according to the staining method is divided into two parts. First, staining method, a very popular staining method, that is a gram staining method. So according to the gram staining method, it's divided into gram positive and gram negative. So gram positive and gram negative. And now we'll discuss about the shape, which types of shape are bacteria have. So shape, first it's a cocci shape, means the round shape, you can say cocci shape. Then rod shape, then spirochete, this is spirochete shape, and finally a no shape, means they can change shape. So we can say it can be changed shape in different bacteria, can be changed the shape. So this is the round shape, that is the cocci. This is the rod, or we can say bacilli. This is spirochete. And this shape is known as pleomorphic shape. Pleomorphic means it can be changed the shape. according to the situation. So that is the pleomorphic shape. So cocci means it can be gram negative and gram positive, means gram negative bacteria which belong to the cocci shape. This is the same thing here, gram negative and gram positive, means any bacteria according to the shape, according to the morphological structure, they are looking like the rod shape, but which it gram negative rod shape or gram positive rod shape. The same thing as spirochete and the pleomorphic, but mostly cocci and the rod or bacillus shape about the gram staining, gram positive and gram negative. But how you will be defined that which bacteria is belong to the gram positive and gram negative? So in the next session we will discuss about the staining method and about the everything about the bacteria, and then we will discuss individually about the bacteria. For example, like the staphylococcal. So a staphylococcal, we will discuss about the species of the staphylococcal, which types of staphylococcal are there, and what is the pathogenesis, what is the laboratory method, what is the treatment, what is the symptom, and all things we will discuss separately every bacteria. So right now just very basic, I would like to say that in a cocci, this is the cocci. For example, it's double circular. This is the diplococci. Diplococci. Then is a long chain. That is the streptococci. Streptococci. Sometime in questions can be there, just mention the diplococci. Means you have to think that which bacteria belong to the diplococci, then you can very easy to answer. The same thing here, some in question that written just long chain series. So you have to know that which types of bacteria belong to the long chain series. And some of them is the cluster form, means like that. It is the grapes like form, the grapes looking like that. So this is a staphylococci. Staphylococci means in a questions you can meet that this types of information that the bacteria is looking like grapes like form. So you can think that grapes like form means it's staphylococci. That's why this is information you have to remember. So about the bacteria, we discussed that bacteria is divided into according to the structure. They divide into two types, typical and atypical. According to the staining method, they divide into gram positive and gram negative. According to the shape, it's divided into cocci, rod shaped, spirochete, and pleomorphic. So this is about the very basic concept about the bacteria. Now we'll discuss about the fungus. About the fungus. Actually, fungus it is the non-photosynthetic microorganism, and it's a acellular, means it's no multicellular, just single cell. It made by the just single cell, and this is the saprophytic microorganism. Saprophytic means it obtain nutrient from the dead cell and decompose organism. Like it can be any dead body, it can be any dead animal, or it can be soil, it can be slime, and even it can be some food. So this fungus is obtain nutrient from these types of organism. And actually it is the sexual and asexual, the both conditions it can be replicate. And the fungus in sometimes very severe infections, severe problems can be there of the skin, like fungal skin and fungus. Even sometimes extracellular organism and intracellular, our internal organs can be damaged, can be in suffering from the fungus diseases. So this is very, we can say, in some conditions very severe conditions can be developed by these types of organism. So about the fungus, this is the saprophytic microorganism, unicellular, and it can be replicate sexually and asexually also. About the protozoa, actually protozoa the same thing, it's also unicellular, and it can be both. It can be intra environment like our internal body, and the external body can be damaged, but mostly internal damage. It is the, we can say that it's mostly found in the intestine, in a liver, in a genital organs, and in some time in a kidney, spleen also. So this is the first of all, it's a unicellular, and it's found mainly in a small intestine and the liver, genital organ, and etc. and so on. And very severe problem can be there. Some like the protozoa, very famous protozoa, that is the Entamoeba histolytica, that can lead to the amoebiasis, and some other protozoa can lead to diarrhea, some other protozoa can lead to some other genital problem. We will discuss right now. Just I am discussing about the introduction of the protozoa. We will discuss very separately about the protozoa, fungus, and bacteria, and every microorganism. About the helminthes and the ectoparasite, they both are multicellular organism, microorganism. Actually, these are the parasite. We can't say this belong to the microorganism, but this is the parasite. Means this is the big microorganism. In some time we can see by my own eye. We can't like without microscope or without any other devices we can see. So helminthes and ectoparasite are multicellular organism. This is parasite. They live in the body, and it can be damage a lot of organ. It can be sucked our blood. So patient, in some patient can be lead to anemia. Actually, the group of the helminthes, it like tapeworm, flu worm, and some other worm is different categories. The subdivided into helminthes in different parts. We will discuss very details whenever we will discuss the helminthes. So right now, just you understand that it's a multicellular organism. A lot of cell. It's made by the lot of cell. That's why the multicellular organism. It's just like a parasitic infection. It's live inside the body and eat our own nutrient, our own blood, our own internal system. So it's very also very danger microorganism. And finally, about the virus and prions. Actually, virus is the acellular microorganism, means they don't have any cell wall. So acellular microorganism, they don't have any cell wall. So actually it's a we can say compose the on protein synthesis, on protein layer for the on protecting by cell. It's by the to the protect by cell. That's why it's compose the protein some protein synthesis. And actually the virus and prions, they will live only in a living cell organism. Means if any organism will die, they can't will live. They will live in living organism and then replicate by self, by the living organism, like a host. They live inside the living organism, and then it's replicate, and it's take nutrient from that body where they're living. So about the virus prions, we will discuss separately when we discuss the virology. So from there you can understand that bacteria means the bacteriology, study of the bacteria. The same thing in a fungus, protozoa, helminthes, ectoparasite, and the virus. We will discuss separately. So first we will discuss about the bacteria, all types of bacteria, everything about the bacteria. Whenever finish the all types of bacteria, then we'll discuss the fungus, protozoa, helminthes, ectoparasite, and virus. So in the next session, we will start the main microbiology. This is the just our introduction about the introduction of the microbiology. In the next session, we will discuss about the bacteria, the staining method, gram staining method, how you will be stain, which types of color will see in the slide that you can identify. This is the belong to the gram positive. This is belong to the gram negative. They about the shape and which types of bacteria are by the like this bacteria is belong to the gram positive and this type of bacteria is a gram negative. And then further we will discuss individually all bacteria. So hope you will understand like the pharmacology, and along with the microbiology and pharmacology, some other important clinical subject and some other important subject that we should discuss, we will discuss. So hope you will understand and like our discussion of the microbiology. Thank you so much.